FLAVR SAVR TOMATO. On May 21, 1994, the genetically engineered tomato was introduced, produced by Californian company Calgene in 1992. Calgene introduced a gene in plant which synthesized a complementary messenger RNA to PG gene and inhibiting the synthesis of PG enzyme. This is the first genetically engineered crop granted license for human consumption.
The first genetically engineered crop that is sold commercially. It is well known around the world because of its firmness whether it is ripe or not. Their expectation was that ripe fruit would remain firm longer, perhaps even allowing it to be transported to market after vine ripening. Transporting vine-ripened fruit would avoid the practice of picking green fruits and artificially ripening them by ethylene treatment.
Gene of interest. In case of tomatoes, PTOM5 gene encodes for phytome synthase, which promote lycopene synthesis that gives red coloration. PTOM6 gene encodes for polygalacturonase. This enzyme degrades the cell wall, resulting in fruit softening. So in this case, GM tomatoes, it promotes good appearance and more taste. So by the help of GM tomatoes, it is good for importation.
In case of tomatoes, PTOM gene encodes for ACC oxidase. This enzyme catalyzes the ethylene formation that triggers the fruit ripening. This explains that a PTOM gene was encoded for ACC oxidase, an enzyme that helps to accelerate or increase the formation of ethylene that causes the ripening of a fruit.
The FLAVR SAVR tomato is defined as any tomato cultivar or progeny of a tomato line genetically engineered using one of the following binary factors, such as PCGN 1557, 1558, 1559, and 1578. The vector system used to transfer the FLAVR SAVR gene to plants is based on the TI plasmid from Agrobacterium tumefaciens. This system is also binary, with the genes to be transferred on one plasmid and the genes encoding necessary functions for transfer, the bird genes, on a second plasmid.
Binary vectors are used in agrobacterium-medicated genetic transformation to generic or transgenic plants. This discovery of using binary system permitted facile manipulation of agrobacterium and paved the way to numerous genetic engineering products, or what we call genetically modified organisms, or GMO.
So this is the mode of transmission of FLAVR SAVR tomato. A second copy of the tomato gene, polygalacturonase, was inserted into the tomato genome in the antisense direction. Polygalacturonase is an enzyme that is one of the most abundant proteins in ripe tomato fruit and has long been thought to be responsible for softening in ripe tomatoes.
The polygalacturonase enzyme degrades pectin, a component of the tomato cell wall, causing the fruit to soften. When the antisense gene is expressed, it interferes with the production of the polygalacturonase enzyme, delaying the ripening process.
Importance, uses, and application of GM tomato. Delayed ripening. The good thing about GM tomato is you will achieve the quality goods or products of your tomato. Second is environmental stress tolerance. So, ang kinagandahan ng GM tomato is even drought or wetness, mga malalakas na hangin ay kayang i-handle ng GM tomato. Third is pest resistance. So, the good thing about GM tomato compared with the traditional tomato is hindi siya madaling dapuan ng peste.
Also, the importance of FLAVR SAVR tomato is improved nutrition and also the taste.